Welcome back to the Scilife Academy. In this session on sterilization, we'll dive into the main concepts related to sterility as well as understanding the terminology involved. We'll start by defining what sterility means and how it is determined, including the use of the probability of nonsterile unit as a measure. Then we'll discuss the importance of viable microorganisms and bacterial endotoxins in the context of sterile manufacturing. We'll also cover the process of depyrogenation, which is distinct from sterilization and disinfection. And finally, we will distinguish between sterilization and disinfection and their respective roles in ensuring a safe and sterile environment. It's a lot to take in, so let's start with a glossary and the fundamental concepts. The term sterility refers to the absence of all microorganisms and pathogens that are capable of growing and reproducing. These microorganisms are known as viable microorganisms. In industries like pharma and medical devices, sterility is an important concept because it ensures that the product is free from any potential contamination and is safe for use. The concept is simple, but it is difficult to establish in absolute terms. In essence, testing cannot guarantee sterility 100%. It has to be assured by a valid, controlled, and appropriately designed manufacturing process. The process needs to be validated and controlled. It is often expressed in terms of the probability of less than the power of -6 of a non-sterile unit. This limit means less than 1 chance in 1 million that viable bioburden microorganisms are present. We'll look at this concept later on. Viable microorganisms are living organisms that are capable of growing and reproducing. Viable microorganisms do not contain microbial byproducts that may cause harm, such as endotoxins. A product is sterile only when there is complete absence of viable microorganisms within it. As you might remember from previous lessons, microorganisms include bacteria, fungi, and viruses in various states, although most microbiologists consider viruses to be non-living organisms. Sterility is an absolute term. A product can be sterile or not. It cannot be slightly sterile or almost sterile. Bacterial endotoxin is the lipopolysaccharide component constituting the outer membrane wall of gram-negative bacteria. These components are particular toxins that are naturally released by living cells and are also released in higher quantities when a cell is broken down. In pharmaceutical production, it is necessary to remove all traces of endotoxin from drug product containers, as even small amounts will cause illness in humans. When endotoxins are injected into the body, they will cause a variety of pyrogenic symptoms, such as an increase in core body temperature, fever, headaches, and nausea. These substances are resistant to many types of sterilization processes. A depyrogenation process is needed to ensure a full destruction of endotoxins. To sum up, a drug product or component may be sterile, that is, containing no viable microorganisms, but at the same time be pyrogenic, because it contains cellular byproducts. Depyrogenation is the elimination of all pyrogenic substances, including bacterial endotoxin, and is generally achieved by complex processes of removal, inactivation, or destruction. It is an important part of the manufacture of pharmaceutical products. It is distinct from sterilization, which refers to the destruction of living cells. Depyrogenation can be accomplished through various methods: heating, filtration, distillation, chromatography, and chemical treatment. In the pharmaceutical industry, depyrogenation is commonly used to sterilize injectable products, medical devices, and other equipment that may come into contact with the bloodstream. The depyrogenation process ensures that the final product is free from pyrogens and safe for use. From the previous definition of sterility, sterilization refers to the use of a physical or chemical procedure to destroy all microbial life, including highly resistant bacterial endospores. Sterilization is a complete process that is able to destroy all forms of microorganisms, whether bacteria, virus, fungi, and spores. It is important to understand the difference between sterilization and disinfection.